Okay, class, we are meeting again, grade 11 life sciences, in regard to revision March test 1. I just want to give you today that we are working on exam revision guidelines and scope. This is scope and exam revision guideline towards end of term 1 exams. So basically what you need to know, class, is that we are having a typical paper here, a reflection of what we're going to write at end of the term. So as you can see, we always have instructions when it comes to exams. Instructions are very important. Please, I always urge you that you should be able to understand and follow instructions whenever they are given. It can be instruction for the whole paper and also instructions per question item. So these are the instructions that are given here. We just go through them, and I'm not going to read through one by one, but I'm just showing you that in any case, when you write exams, instructions are very important. We go straight to question number 1, which I want you to understand that answers that are given here, they are more or less correct, but we want the most correct one, the most accurate one, with amongst the given options that are there. So we are given now that which of the statements below represent a similarity between barophytes and petrophytes. We have talked about classification of plants. I want you to understand that when we are looking at these, we are looking at plant kingdoms. I guess this has been explained again. If you go to our channel, it has been explained. Just to recap for some of you, we have not yet connected, or we are just connecting to Metric Hub for the first time. We are saying uh, barophytes are non-vascular plants, which we say that they do not have the xylem or phloem vessels, which I said last time that these are vascular plants, or these vascular tissues that we are talking about. They are responsible for the transportation of water and nutrients within a plant. So we need to understand that the barophytes, they basically don't have vascular tissues, whereas the petrophytes, they do have. And then we have got quite a lot of other simple small, we are saying they are small, simple, and usually grow in damp areas. These barophytes, they are quite simple and small. They normally grow in damp areas. We have got examples of mosses and wormwood. These are typical examples of barophytes. Again, we need to know that the petrophytes include ferns and their allies. So if you can check in the method of reproduction, you shall see that the barophytes also reproduce via spores. They reproduce via spores, and of course the petrophytes also they reproduce via spores, but they do have complex structures. So what I want you to understand is that exam will always come, and then you need to know that answers which are given, they will be more or less correct, but the question wants you to give the most correct answer there. So now, with the given options, we can agree that both division reproduce by means of spores. They both reproduce by means of spores, as I have just mentioned there. They both reproduce by means of spores. Uh, and you remember we said that the barophytes, they do not have vascular tissues. Of course, these ones, they are there for what in petrophytes plants. Petrophytes, they do have vascular tissues. However, the barophytes, they don't have the xylem and phloem vessels. They don't have them. So the answer is B. We go to the next question. We are given the diagram of a plant or of a flower. We need to understand, like to say, where does pollination and fertilization take place? Where do we have pollination and fertilization? We know pollination is the transfer of pollen grains from the anthers to the stigma, and then it is thus it. We are talking about the carrying of the seeds, and then fertilization is the fusion, whereby to say Now fertilization now it looks into the fusion of the male and female sex cells in order for them to produce a new flower or a seed. So now we are given number question now, question 2, where do we see pollination and where do we see pollination and fertilization occurring? So we should understand that in terms of this, where does pollination and fertilization occur? We should understand that pollination it takes place to the stigma. It takes place to the what? We are saying we are looking at pollination. We are seeing that question there. It has to be X where pollination occurs, and then we have got fertilization occurring. Fertilization it occurs in the ovules, which is Z. We have got fertilization occurring, fertilization, and then we have got fertilization occurring there, and then we have pollination occurring by the stigma, which we say that the pollen tube or the pollen grain, it has to move through the stigma, the style, and then it moves down to the ovule, or where it has to, where fertilization must occur. So we are saying the answer is what is C, the part of the flower which becomes the fruit. I've been giving this also on the channel that you need to take note of that to say which part of the flower develops into the fruit. We said developed into the seed, developed into the fruit, develops into whatsoever. So now what you need to know is that the ovary develops into the what? It develops into the fruit. It develops into the fruit. And then which is the ovary, and then give the correct biological term for each of the following descriptions, right? Only the text next to the question number. So reproduction, which uses the non-reproductive parts of a flower to produce new plants. We say that we have got two types of reproduction processes. We have got sexual, and we do have also as number one, and also we have what is called asexual. Types of reproduction, sexual and asexual. So asexual is where we talk about vegetative propagation. Vegetative propagation. What happens here? We are saying there is no seeds, no seeds are used. This one, sexual reproduction, it looks into fertilization. It looks into fertilization. It looks into seeds. And then if you can understand very well, what you need to know here is that the reproduction, which uses non-reproductive parts of the plant, it is called what? Asexual reproduction or vegetative propagation. It goes vegetative propagation or asexual reproduction. Asexual reproduction, which we are saying we use some parts of the flower itself. Adult plants, adult plants are used to produce new vegetation and so forth. So now it is asexual, meaning to say that there is no fertilization that occurs there. And then symmetry found in the phyla, it is actually asymmetry. We talk about asymmetrical, which is number 2. It goes we say that it has no symmetry, so it is called asymmetry. And then a diagram showing the evolutionary relationships between organisms. We can be able to trace the production or the evolution of organisms through what we call a cladogram. A cladogram is used to represent or to show the generations that have successively developed from a certain or within a certain species. So that is called a cladogram. It is called a cladogram, and others can call it. It is called a cladogram. Others can call it a phylogenetic tree diagram. A phylogenetic tree diagram. Phylogenetic tree. It actually establishes, it identifies the relationships that have developed within a population species, tracing where the population started and how it is growing. So you need to know that that is called what? That is called a cladogram or a phylogenetic tree. We use it in understanding evolution. Evolution is understood through that kind of illustration, seeing which one extincted first and where this other species developed from, and otherwise also identifying species of organisms that lived within the same era or same time. So we need to go to question number 1.3, which we are supposed to identify the statements in column A applies to A, B, both or none with from B, from column 2. Write only the letter next to the question number. So we are saying gametophyte is dominant. We see that gametophyte is dominant in terms of the mosses. The mosses are dominant with the gametophyte. So we are saying B, naked seeds. Naked seeds, these are found also mostly in the gymnosperms. These are also found in gymnosperms, which we say that it is B again. And then the highly modified floral leaves, which make up the pistil of the flower. The pistil of the flower is made up of the carpels. The carpels are there to make it is there to make uh, the pistil of the flower, which is a only B and B. So we need to note that these are the answers with reference to the question that is given here. So section A, 15 marks, they are possible to be collected, all of them. There is nothing complicated here. It's a matter of just understanding what the question wants before you start answering. So now we are given section B, the diagram showing plants from different divisions of plant kingdom, the different plant kingdoms, the plantae kingdoms, or what we call the different plant kingdoms. So now we are given A, B, C, and also we have got 4, which are numbered from 1 to 4. Identify plant division 1 and 2 respectively. The plant kingdoms. Please, again, I say refer to channel in terms of the full video that explains this. We are not gonna go through detailed explanations again because of time. So we are saying identify division 1, 2, and 1 and 2 respectively. So 1, if you can see, this is the petrophytes. The petrophytes. 1, it is called the pentophytes, the petrophytes, and then 2 is the barophytes. 2 is the barophytes, the two divisions of one and two, the petrophytes and the barophytes. I guess I have already explained the differences that we also answered when we answered question number one. State two difference between one and four with the reference to reproduction. One and four, we are saying two differences between one and four. One, in terms of one and four, one, it actually, if you can look there, it is a plant which we say the petrophytes, it actually need water for fertilization. And then four, if you can see four, it actually does not depend on water for fertilization. One depend on water, one depend on water, one it depend on water for fertilization. Well list, we see four does not need water. It does not need water for fertilization. And then we can also say something again which has to do with uh, number one. As you can see, normally we can agree that with number one does not have, it actually have no seeds. One, one have no seeds. One, it have no seeds. And then four does have seeds. We are saying these are the major differences that we can give with regards to question number 1.2. Question 1.2, we are saying, if you can look at it, you know that question plant A or plant group, we are saying group one, it actually they need water in terms of their reproduction or in terms of their fertilization, where list four does not need any water. It doesn't need water for fertilization, which we can see that that is one key difference that we can mention. And then one again have no seeds. As you look at it, it doesn't have seeds, and four, it does have. And then question 2.13, question 2.13, if you can look at it again, it says state only the number of the plant group which has no vascular tissue. The one which has got no vascular tissue, as you have seen there, we said that number we said the plant that do not have the vascular tissues. We already mentioned that these are the barophytes. The barophytes do not have vascular tissues, which is plant group one. I mean group two. Group two do not have vascular tissues, so it is group two. Group 2 do not have vascular tissues class. The barophytes, they don't have the vascular tissue. That is why we also explained that in the beginning when we defined the petrophytes and the barophytes. So barophytes is two. They don't have vascular tissues, the xylem and the fuel and fossils for the transportation of water and nutrients. So we are given question 2.2 here, showing various views of the board plan of an animal belonging to the phylum Plantem. So now what we need to understand is again that when it comes to this, you need to know basically to say uh, when we are talking about this type of animals, we say that these include flatworms, which we can give, such as the tapeworms and the liver floats. They are actually soft-bodied organisms, often also parasitic. They are often parasitic, these types of animals. They are also parasitic. We are saying they actually have examples such as those of tapeworms and the flatworms. And one other thing is that they have no simple nervous system. They have a simple nervous system. Some are free living, while others infect hosts like humans in order for them to survive, which can we can give examples such as the tapeworm and the planarians, which we can give as examples of the type of animals belonging to this group of animals here. So now we are asked to say that we have got, for example, the achines, and then they are surrounded by the ganglia and the central lobes, the nerve cord. I guess you have seen this again. You shall see it as we go to evolution, as in terms of the similarities of organisms and so forth. So now the question is saying, provide labels for the germ layers A, B, and C. A, B, and C. We are saying that A, it is the outer layer, which is called the ectoderm. A, it is the ectoderm. That is A. And then B, we need to know that is the mesoderm. B is the mesoderm. And then C is the endoderm. Those are the three layers that we are given there. A is the ecto, and then we say B is the meso, and then we have got C, which we say is the endoderm. This question is also quite common most of the times that you find in life sciences. We quite a lot of times have to answer that one and then explain one way this animal's body plan is suited for its mode of life. If you can look at it, we are saying these are also type of organisms or animals that we can say that they are dorsiventral differentiated, since most of them, or since it is mostly free living worm live at the bottom of mud waters, and so you have their eyes on their upper surface. Their eyes are on the upper surface, as you can see there. That is one good thing that you need to know. One, just say one. Uh, they are actually we are saying they are also ventrally differentiated. We see most of them are free living flatworms. free living flatworms, which like live in the bottom of mud waters. They live at the bottom. They live at the bottom of mud waters. Hence you find out that hence their eyes, you find that their eyes are on the upper surface. That is question, that is one. And then we can also have two, which we can also understand that these also these organisms or these creatures, we can also say that they have civilization. They have civilization. Civilization. They do have first civilization as they come into contact with new environments. As they come into contact with the new environment. So that is one thing. We also want to understand that with new dangers and food sources regularly, so there needs to be what? To be an accumulation of sense organs at the part that enters these new environments. First, they have civilization as they come into contact with new environment, which I want you to understand that with new dangers and food sources. With new dangers and food sources. So I want you to know again that uh, we need to understand that they have got an accumulation of sense organs at the part that they enters into new environment for the first time. New organs are also developing as they enter into new environment for the first time. And then other point that I want you to notice that these organisms, the development of the mesoderm or the triboballistic allows the development of muscles in these organisms, which allow free-moving flatworms to actively move from one place to another. It is because of we are saying the development of the mesoderm and the triploblastic, uh, which allows development of muscles and also free living flatworms to develop or to have the ability to move from one environment to another. So next question, it says tabulate two differences between the phylum Plantimides and then also the phylum Cydrea. So now what we need to see is that the difference is that we can, when they say tabulate, it means they want you to put them in a table form, which we can give to say uh, we draw a table here. Draw a table there, and then we have got the Cydrea. And then we want to give the differences. So we can say one we say these ones, we say there's civilization. Civilization occurs. A civilization occurs, and then in this case, there is no civilization. Two, we can also talk about this. They have got the dorsoventral differentiation. They have a dorsoventral differentiation, which I want us to understand that in terms of the other class, you need to understand that there is no, it's not there. You don't have it. Number three, we might also need to talk about the bilateral symmetry. And this other one, they do have a radial symmetry. We have a bilateral symmetry. And then we do have a radial symmetry. Radial symmetry occurs. I think this can be major difference that we can give. But of course, maybe to finish up, which I can give as the fourth difference, we have got the tripoloblastic. And then we talk about the dye. So we are saying here we talk about three, and then the other one, it has got what? We are saying we are talking about the tri, which means three. Uh, we have got two. That is the difference, which you must always be able to note as you go to exam. Please take note of this. We have got question next. You can just have to do question next first. It's a group of grade 11 learners investigated the effect of temperature on bacterial growth and used the following procedure. 5 petri dishes containing agar. Actually, they were labeled with corresponding incubation temperature, and then the incubators were set at different temperatures, for example, 10, 20, 30, 40, and 50 degrees Celsius. After sterilizing the inoculating loop using a Benson burner, It was dipped into the bacterial culture and gently moved across the what? Across the surface of each agar plate, ensuring even distribution. The learners inverted the plates and placed them in their respective incubators at the designated temperatures. After incubation, the number of visible bacteria colonies on each plate were counted. So we have got the temperature that is given here, and then we have got also the number of colonies that also get in there. So we have got the question which says identify the independent variable in this investigation. Identify the independent variable in this investigation. So now we need to understand is that we are looking at an investigation that has to be looked on the number of colonies that can be established. So now we are saying effects of temperature, the effects of temperature on bacterial growth. The growth of bacteria is affected by temperature. Growth of bacteria is dependent on temperature variation. It is based on the temperature differences that is going to happen there. So now I say that when you look at an investigation, you always need to understand that the independent variable is found when we talk about an investigation was done to what. So now we see that the independent variable in most of the cases, it has to be the beginner when we talk about effects of it is temperature that is affecting bacterial growth. So the independent variable is temperature. It is temperature, which is an independent variable, and the growth of bacteria is the dependent anyway. Dependent is what we say dependent. We look at that as the growth of bacteria. The growth of bacteria is affected by temperature. So now that is how it goes. So our answer is temperature, class. Our answer is temperature. And then that is temperature in terms of first question. Temperature, if we look on the first question, temperature is the answer there. Give one reason why this experiment may be considered unreliable. There are certain preparatory or there are certain practices that must make an experiment reliable. Reliability of results is affected by many factors. We can talk about repetition of the investigation. Uh, we can talk about the repetition. We can also talk about the increase in the sample size. We can talk about the maintenance of environmental conditions across the settings, the setup points. So one, we are saying experiment was not repeated. The experiment was not repeated. It was not repeated, this experiment. So therefore, we cannot actually base on the results on that. There is no repetition of the investigation. And then we can also say that the sample size was very small, which is seen by only one petri dish that was used. Only one petri dish was used uh, in terms of temperature treatment. only one different temperatures are sterilizing the Bunsen burner was used. But again, we said that the learners did all what they did in the end. We should know that we should also know that the sample size was very small. It was very small, which you can see that only one petri dish was used for temperature treatment. Only one petri dish. Only one petri dish was used. It was used for temperature treatment. And then identify one way in which the learners could have increased validity in this investigation. We can talk about in this investigation how learners could have increased their validity. We talk about they should have used same. They might have used the same type or a quantity of the what of the agar. And then two, we might also need to talk about can also talk about same type or quality of this agar, which we need to know that it can make the result to be valid, and then use the same species of bacteria. Use the same species of bacteria. It would also mean that we are going to have results that are very correct because we did not change the type of organisms or the species of organisms that was used. And then we can talk about uh, the incubation period could have been the same. Use the same or have the same incubation. It must be the same incubation time. We are talking about the duration in this case. How much time was used there? It must have been the same. And then question 1.4, explain why inoculating loop was sterilized before dipping it into the bacterial culture. The idea of sterilization is actually to kill germs, so to ensure that no other bacteria are introduced into the agar except the bacteria used in the investigation. It is actually for destroying the other bacteria that might be there, so to ensure no other bacteria are introduced. No other bacterias. No other bacteria are introduced. That can be correct to say that we wanted that when you look into the agar, we want only to accept that the bacteria used, we want to ensure that the bacteria used in the investigation is the only one that must affect the results. So we are saying to ensure that no other bacteria are introduced, such that uh, are introduced, which we say except the bacteria on the bacteria that was introduced, except the bacteria that was introduced. So we now need to worry about a paragraph to show the relationship between temperature and growth of bacteria. We are saying temperature and the growth of bacteria is what we are going to work on. We have got temperature and then a number of colonies. So now the question is saying draw a paragraph that must show this. So what we need to worry class about is that the graph should always have a title. A title must always be given when we talk about a graph. So now we are saying temperature against the number of colonies that are there. So we can say that we want to look on effect of temperature on bacterial growth, that we need to understand effects of temperature on bacterial growth. Effect of temperature. It is temperature that affects the growth of bacteria. So now we are given that what we need to know is that the number of colonies is the one that is defining the number of colonies is the one that is defining the shape of the graph. So we said the independent variable goes on the horizontal axis. I'm sure that we have talked about that severally in terms of the issues of dependent and independent variable. So we can say that temperature, we said, is independent, so it has to be on the horizontal axis. We have to say we have got temperature on the horizontal axis, and then we have got number of colonies this side. Number of so now we have got our graph going up there, and then we have got temperature. We have temperature. Temperature is on the horizontal axis. So we are going to look on temperature and their corresponding number of colonies that are there. At 10, we have 10. So we are saying we need to look on the number variation, the smallest versus the biggest, so that we can choose a suitable scale. A proper scale must be selected. So we have got from 0, 10, 75, 250, 100. So therefore I can choose to use a certain scale, which I can say I can use a scale of if I use a scale of 100, if I use a scale of 100, I think that can also do a scale of 100. So I do have 100 on the lowest, 100, 200, 3, 4, 5, 6, 7, going there. So we have got 100, we have 200, we have 300, we have 400, 5, and therefore 7 on those axes as I've drawn there. So at 10 temperature, we have got 10. At temperature 10, we can say we have got 10. That I can give 10 is the We have 10 here. And then we have got 10. And one thing that I want you to take note of is that a bar graph, we can draw it without the bars touching each other. So we have got 10, and then the next one is 20, is 75. 20 is 75, which is above this one, but closer to 100, but not 100. So we have got 75 there. We have 20. And then the next one we have got is 10, 20 up to 50. It's 30 is 30 is 250. 30 is 250, which we can say our 250 is above 100, I mean above 200, but between this is 250, this is 250. So I always say that the graphs, you can color them in order for them to be smart. So we have got 40. At 40, we have got 100. At 40, we have got 100. At 40, we have got 100. And then we said 40 is 100. And then lastly, 50 is 0. 50 is 0. So our graph comes in like the way we can see it here. That's how the graph has to be like. So if we do our graph like this, we are saying temperature versus number of colonies. We are saying temperature, which we can put here again. And then we say number of colonies is there. That's what happens with our graph. A graph must have a title, which is given there. A graph has a title, yes. The title is there. We are given title there. And then the axes are labeled. We have got the number of colonies, which is also given in this case. And then we have also temperature on the horizontal axis. And again, we said we have got readings that must be there on the vertical axis. So we have got some key that is correct. So we can say that our graph is drawn, and that's how it has to come up like. That's how it has to come up like. So we have drawn the graph effects of temperature on bacterial growth. That's the title which we have given our graph, which is quite correct. And then we want to go to the next question. It's saying study the diagrams below. Provide labels for 1, 2, and 3. For 1, they're saying 1, 1, 2, and 3. Provide labels for 1, 2, and 3. So 1, you can see that is the flagellum, that is 1. 2, we said 1, 2, and 3. 2 it can be the nucleus, which we can say that it can be DNA, it can be RNA. And then 3 can actually be, this is asparagorium. These are the labels that we have given there, very correctly, and they are correct. And then read the extract which is given below. Malaria is deadly disease caused by Plasmodium SP and transmitted through the female Anopheles mosquito. In 2020, 627,000 people around the world died from malaria. DDT is a pesticide used to control malaria populations in malaria areas. In the early 1990s, there was a worldwide ban on the use of DDT. DDT caused the decline of many birds of prey as it made the shells of their eggs very thin. It affected the birds in reproduction. That's what is said here. The number of deaths due to malaria rose from 1991 to 450 in 2000. The South African government decided to lift the ban and started using DDT again. It's, and then by 2020, there were only 38 deaths due to malaria. So question number 3.3, it says, explain how the use of DDT caused the decline in many birds. It is given that it actually affected their shells. It made the shells of their eggs very, very thin, which caused difficulties in hatching. So shells of their eggs became very thin. Shells of their eggs, shells of their eggs, they actually became very thin. They became very thin, making the eggs easy to break. It made the eggs very easy to break. It made the eggs very easy to break because the shells they became very thin. That is what is the effect is on this one. Calculate the percentage of deaths. Calculate percentage of deaths to malaria in South Africa 2020 compared to the number of deaths from malaria globally. So we just need to look at the percentage which is given there. We need to look into the extract, which says that we need to look at this one. The population statistics are given. We are told that the population by 2020 there were only 38 due to malaria. So now what we are saying is it is the total 627,000 people around the world died in 2020. Around the world, people died, which is against the number which is given there, that 38, only 38 people died. In the other times, 38 deaths only, 38 deaths. So in order to calculate percentage is 38 This is equals to 38 divided by 627,000. We say that multiply by 100 because we want to calculate a percentage which can give us which we can give as to say that it can give us 0,006 0,006%, which we can give to two decimal places, which is equals to 0,001. This is according to two decimal places. It can give us what? 0,01 percent. 0,01% to two decimal places. That can be correct. So class, questions such as these ones are just bonus marks, which I always expect you guys that you don't miss it and you don't get problems. It's a matter of just identifying facts that are there and then relate them to the questions so that you can get good marks and good answers. My pleasure for having you following Metric Hub. Please don't use your don't miss NSFAS, which is our national funding platform or funding support. Connect to Metric Hub right now so that you won't have any mistakes. Thank you very much and keep connected. Please don't forget to subscribe, like, and also to join membership for you to become a full-time member of the group. Keep connected. Thank you.